When we talk about clinical microbiology, you'd be hard-pressed to find an organism as fascinating or as frustrating as Escherichia coli, right? It's a quiet resident of our gut, but it's also a pathogen that can be incredibly dangerous. In this explainer, we're going to follow its story all the way from the culture plate to the patient's bedside to really get a handle on this duality. And this quote just hits the nail on the head. It perfectly captures that central theme, that core challenge of E. coli. It really does have two faces. On one hand, it's a vital part of our normal gut flora. On the other, it's this incredibly adaptable pathogen. And that dual nature, well, that's at the heart of everything we're going to cover. Just so you know, all the information we've pulled together for this explainer comes from a Gemini deep search. If you're the kind of person who loves to go down the rabbit hole and check out the primary literature, the full list of sources is right there for you in the accompanying notebook document. Okay, so here's our game plan. We're going to start with its genetic secrets, what makes it tick. Then, we'll hop over to the lab and look at its profile. From there, it's all about the diseases it can cause, the huge challenge of resistance, and finally, how we manage it all in a clinical setting. Let's dive right in. All right, Section 1, a Bacterium of Duality. We're kicking things off by looking at its taxonomy and, more importantly, the incredible genetic plasticity that really defines what this organism is and what it can become. Now, this is where it gets really interesting. This visualizes the secret to Escherichia coli's incredible versatility. You see that small slice? Only about 20% of its DNA is the stable core genome, the essential housekeeping genes that every Escherichia coli needs to survive. But that other 80%? That's the pangenome. Think of it as a massive fluid library of optional genes that can be swapped between bacteria. And this is where all the nasty stuff, the virulence factors, the resistance genes, lives. It's what makes each strain a unique potential threat. And for a really striking example of this, look no further than Shigella. Genetically speaking, the 4 Shigella species aren't really a separate genus at all. They're basically just specialized pathivores of Escherichia coli that happen to pick up a specific invasion plasmid and evolved to cause dysentery. Now, for clinical and historical reasons, we still call them Shigella, but it just goes to show you how blurry the lines can get with this organism. So how does all that genetic chaos translate to what we see in the lab? Let's move on to Section 2, the laboratory profile. We're going to walk through the practical, day-to-day steps for identifying Escherichia coli. First up, the good old gram stain. For any microbiologist, this is a familiar sight. Escherichia coli has that classic morphology. It's a gram-negative straight rod. You'll usually see them as single cells or maybe in pairs. Most strains are motile, and a key point, it's non-spore forming. Pretty straightforward. On the culture plates, its appearance is just unmistakable. On McAconkie agar, it's a vigorous lactose fermenter, so you get those bright pink colonies. But the real showstopper is on EMB agar. The acid production from lactose fermentation is so intense that it actually causes the eosin and methylene blue dyes in the agar to precipitate right onto the colonies. And that's what creates that classic, iconic green metallic sheen. You see that? You're thinking Escherichia coli. And to really lock in the identification, we turn to the biochemicals. The classic IMVIC test series gives us that definitive footprint. For Escherichia coli, the result is just rock solid. Positive, positive, negative, negative. That's indole positive, methyl red positive, Vosges-Proskauer negative, and citrate negative. That plus plus minus minus profile is a cornerstone of its ID. Okay, moving on to Section 3, diverse clinical syndromes. Now, we're going to bridge the gap from the microbiology to the actual patient, taking a look at the 2 major pathogenic divisions of Escherichia coli. So the first big split is all about location, location, location. You've got your intestinal pathogenic E. coli, or IPEC. These are the guys that cause disease inside the GI tract, think diarrhea. Then you've got the extraintestinal pathotypes, or XPEC. These are the strains that start as commensals but then escape the gut and cause trouble in normally sterile sites, like the urinary tract or even the central nervous system. Zooming in on that intestinal group, different virulence factors lead to very different diseases. For example, introtoxigenic Escherichia coli, ETEC, and its toxins cause that classic, watery traveler's diarrhea. Introinvasive Escherichia coli, i.e. IEC, acts a lot like Shigella, invading the gut lining to cause a bloody dysentery. But the one we really worry about is Shiga toxin-producing Escherichia coli, STEC. Its powerful toxins can cause not just bloody colitis, but also the life-threatening hemolytic uremic syndrome. And this brings us to an absolutely critical clinical pearl. For S-tech infections, giving antibiotics is a big no-no. There's good evidence to suggest that killing the bacteria can actually trigger a massive release of Shigella toxin. This can kick off or even worsen hemolytic uremic syndrome. This is one of those things you just have to know for proper patient management. The threat of a disease like HUS really underscores the need for rapid, really informative diagnostics. So, that brings us to Section 4, Resistance and Molecular Diagnostics. Let's look at the ever-growing challenge of resistance and the modern tools we have to stay ahead of it. This is really the central question for any modern microlab, isn't it? As resistance continues to spread like wildfire, our diagnostics have been forced to evolve. It's not enough anymore to just know what the organism is. We have to know its resistance profile, and we need to know it fast. And this is how we're doing it now. The modern workflow often leans on molecular methods to completely bypass those time-consuming culture steps for that critical initial decision. We can go straight from a specimen, a stool sample, a positive blood culture bottle, to a multiplex PCR panel. And these panels are amazing. They can detect the pathogen and screen for key resistance genes, like ESBLs or carbophenamases, all at the same time. This gives clinicians actionable results in just a few hours, not days. It's a game changer. Now, of course, phenotypic testing is still the gold standard for getting that full antibiogram. And here are the latest clinical breakpoints from EUCAS for interpreting the MIC, or minimum inhibitory concentration. So for meropenem, an MIC of 2 or less is susceptible, while anything over 8 is resistant. These standardized numbers are absolutely crucial for guiding targeted, effective therapy. Which brings us to our final section, clinical management and control. Let's take all this benchtop knowledge and apply it right at the bedside, talking about guideline-driven treatment and, of course, prevention. Treatment guidelines have to be tailored to local resistance patterns. And this slide showing the Dutch swab guidelines is a great example. For a severe UTI with systemic symptoms, they recommend an IV third-gen cephalosporin. For simple cystitis, it's oral nitrofurantoin. But look, the duration is different for men versus pregnant women. It's always interesting to compare this to, say, the IDSA guidelines in the US, which also lists fosfomycin as a first-line option for uncomplicated cystitis. It just reflects different regional pressures. Okay. This right here is a major practice-changing recommendation. For bloodstream infections that are caused by third-generation cephalosporin-resistant Enterobacterales, or 3GCRE, you need to avoid piperacillin tazobactam. Why? Well, the landmark Merino trial showed pretty clearly that for these specific bugs, patients treated with pip/tazo had a significantly higher mortality rate than patients who got a carbapenem like meropenem. It's a huge deal. Treating kids, as you know, requires a totally different playbook. For gastroenteritis, the #1 priority is oral rehydration therapy, not antibiotics. We have to say it again. For suspected STEC, both antibiotics and antimotility drugs are contraindicated. For UTIs, dosing is always based on weight, and we generally avoid entire classes, like fluoroquinolones, because of concerns about potential cartilage damage or arthropathy in growing children. And finally, let's talk prevention. To stop pathogenic Escherichia coli from spreading, especially the multi-drug resistant strains, basic infection control is everything. It's not fancy, but it works. This means getting that patient on contact precautions immediately. Single room, gowns and gloves for everyone entering, meticulous hand hygiene, preferably with soap and water, and using dedicated patient equipment whenever possible. So we'll end with a question to chew on. Given this organism's incredible genetic plasticity, what's the next scary hybrid Escherichia coli pathotype we're going to see? I mean, that EHE-EAC hybrid strain in Germany back in 2011 was a terrifying wake-up call. What combination of virulence factors do you think is coming next? Let us know what you think in the comments. And hey, if you found this explainer helpful, please hit that subscribe button. Thanks for watching.